Hello everyone, in this lecture today I'm going to talk to you about what is TCID50 assay, what is the protocol to perform this TCID50 assay, and how we can make the calculation for this TCID50 assay. Okay, so first, what is TCID50? As the name suggests, TCID50 is the 50% tissue culture infectious dose 50. Okay, so this is tissue culture infectious dose 50. This is an assay that is used for the quantification of virus. Okay, quantification of virus that produces cytopathic effect in 50% of the inoculated cells. Okay, so this is the TCID50. So basically, what is this assay? This assay is used for quantification of virus. Okay, so this assay is used for quantification. Okay, so this is used for quantification of virus. Okay, quantification of virus. So this is TCID50 or viral titer. Okay, so this TCID50 is an endpoint dilution assay that is used to quantify the viruses that do not form plaques, because we also have another assay called plaque forming assay for the quantification of virus, but this assay is used for the viruses that do not form plaques. Okay, so in brief, what we do in this TCID50 assay, serial dilutions of the virus are added to the host cells. So in the cells where we want to perform this TCID50 assay, what we will do, we will add the serial dilutions of the virus to the cells, and we will incubate these cells with the virus. And after the incubation, what we will do is that we will calculate the percentage of the cell death. Yes, will be measured, not calculated. This is measured, but how? From a cell viability assay using a plate reader, we can measure the percentage of the cell death using a plate reader, or alternatively we can also use microscope. Okay, so cytopathic effect in short forms CPE. Okay, so then the dilution at which 50% of the cell cultures are infected, which is the endpoint, is used to actually mathematically calculate TCID50. Why this assay is called result and is expressed as 50% infectious dose TCID50 per milliliter. The reason why this assay is called TCID50 is because the dilution of the virus at which 50% of the cultured cells are infected. Okay, so we use that dilution to make the calculation for this assay. That's why it is called TCID50. Okay, that is the reason why we have this 50 in TCID. Okay, so TCID50, right? So this is the introduction. So basically, in synopsis, it is the assay that is used for the quantification of the virus, and in this assay we make the calculations at which 50% of the cells are infected with the virus. So that dilution is used to calculate TCID50. Okay, so then what is the protocol? So the protocol is that, first let's take an example. Let's say that we have A6 cells. So what we will do, we will plate these A6 cells in 96 well plate. I'm also giving here the number, there are 5,000 cells per well for about 2 days in 100 microliter media. Okay, so we will seed the cells for 2 days in this 96 well plate. This is point number 1. And in number 2, then what we will do, in a separate plate, that is a V-shaped plate, 96 well plate, we will make the dilution of the virus. Okay, so I mean the virus for which we want to determine the quantity of, right, the virus whose quantity is to be determined. So we will make the dilution of that virus. Okay, it can be supernatant from the cells infected with the virus, or simply the virus suspension. Okay, so we will make the dilution here. In this example, we will take 8 microliter of the virus, 192 microliter of the media, 1:25, and then we will make the serial dilution. Okay, so what we have to do, we have to do the serial dilution, right? Serial dilution. We have to prepare the serial dilution of the virus. So first, what we will do, in, let's say that in A, what we have 8 microliter virus plus 192 microliter media. And from here, okay, this is what we have in A. And in B, what we will do, we will take 60 microliter of this. Okay, we will take 60 microliter from the wells in A, and then we will put 60 microliter media, right? 60 microliter media, okay, plus 60 microliter media. Yeah, in this way we will make the serial dilutions here, and this is the schematics, right? So we have to make the serial dilution of the virus, right? And after making the dilution, after we finish making the dilution of the virus, before we are going to the next well, so we have to like resuspend the wells really well, okay? Resuspend the wells very well, okay? So this is really important, okay? So resuspend the wells, resuspend the virus particles in the wells well before going to the next dilution, really important. And so what we will do then now, then we will transfer 100 microliter each of the dilution to the plate with the A6 cells, okay? So now here, okay, we made this dilution in this V-shaped well, this well, right? This well plate, sorry, in V-shaped well plate. So then this dilution, 100 microliter each of the dilution, will be transferred to this A6 plate. So now we will transfer this dilution to this A6 plate. A6 plate, that means the plate in which we have the cells already seeded, right? So for this example, we have A6 cells. To make a total volume of 200 microliter per well, because we had here 100 microliter and we're transferring 100 microliter from here, so it will make 200 microliter per well. So, but we will leave column 1 and 2 without the virus, because these will be controlled here. So in column 1 and 2, okay, so column 1 and 2 actually we will leave without the virus, so these will act as the control, okay? So then after this, after we have inoculated our cells with the virus, then after inoculation of cells with the virus, we will leave them at 37 degrees Celsius with the CO2 supply for approximate 5 days, and we will check them daily. But I have to mention here that the number of days also depends on the cell type, okay? So now after this, then now comes the calculation. So how are we going to make the calculation? So we will check in the microscope, or we will determine, okay, so by checking in the microscope, because here the way we can do is from the cell viability assay using a plate reader, or we can check in the microscope, right? So then we will determine, okay, at which dilution what percentage of the cells are dead. For example, this is 10 to the power minus 1, this is the first one. So when at this dilution we have 100% cell death. In this another dilution also we have 100% cell death, another dilution also 100% cell death, 100% cell death, 100% cell death, and here 80% cell death. Okay, this can be cell death. So how will you know that? We will buy the plate reader, or by using the microscopy we will determine this, okay? Or in place of percentage cell death, it could be percentage infected cells, okay? Percentage infected cells, it could be that, okay? So now we will have this calculation. So at 10 to the power minus 7 we have none of the cell death, and also at 10 to the power minus 8 we don't have any cell death. So then how are we going to make the calculation? So first, what we will do is that we will calculate proportionate distance PD between the two dilution in between 50%, because you remember from previous slide, the important term is the 50%. So we will see where is the 50% coming. So this is 0% and this is 80%. So in between these 2% is the 50%, right? So then how are we going to calculate the proportionate distance between the two dilution in between for 50% death? So what will be that? So proportionate distance will be percentage next above 50%. So basically percentage next above 50% is 80%, because here we have 80% cell death. This number could be 70 or what, it could have been different for different cases, right? But for here we have 80%. So percentage next above 50%. So you have to, here really important, from between 0 and 80% comes 50%, right? So percentage above 50% is 80% here in our example. So it will be 80% and minus 50%. This is from the formula. Okay, this is the formula. So minus 50% divided by, so this is the division, divided by, and then we have percentage next above 50%. So basically percentage next above 50%, immediately next above 50% is 80% minus percentage next below 50%. Okay, so below 50% is this 0%. Okay, it could have been like 20% also. Yeah, but here in our example, because none of the cells are dead in these wells, that's why we have 0%. Okay, so you have to adjust accordingly. So then proportionate distance will be 80% minus 50% divided by 80% minus 0%, because percentage next below, here we have 50% and below 50% we have 0%. It could be different for different cases. Okay, so this is, and when we do this calculation, then we will get 0.375. This is our proportionate distance. And so then what we have to do, we have to calculate 50%, calculate 50% endpoint. That means log lower dilution. What is the log lower dilution? So point number 2 is dilution in which position is next above 50%. Okay, so basically 50% is here, and the percent next above 50% is 80%. And if you go here, the dilution is 10 to the power minus 6, because this is the dilution, right? So dilution is 10 to the power minus 6. So dilution in which position is next above 50%. That means log lower is 10 to the power minus 6. How come this came? Because the 50% just next to 50% is 80%, and corresponding dilution is 10 to the power minus 6. Therefore, we have log lower, which is 10 to the power minus 6, is actually minus 6, right? So this is how we have this number. And then what we have to do now, here we have calculated PD, which is 0.375, and here we have calculated log lower, which is minus 6. Then what we will do in this step 3, we will add PD and log lower dilution. For example, that will be 6, of course minus sign, plus 0.375. So, and it will be 6.375. Okay, you will not do like normal mathematical calculation, just forget this minus here. So 6 plus 0.375 is minus 6.375. Okay, you will not do the normal math calculation, that you have to remember. Okay, so not minus, you will forget minus while making this addition, and afterwards you will put this minus sign here. So we have now 6.375. And then, so after this, so to calculate the log TCID50 will be 10 minus 6.375, which is actually a 2.37 into 10 to the power 6. So basically TCID50 is actually 2.37 into 10 to the power 6. But this is 2.37 into 10 to the power 6 is our 2.37 into 10 to the power 6 is our TCID, but we have to calculate TCID50 per ml. Then we have to divide. So divide by the ml of virus inoculum added to row A. So basically how much we added, you remember the original virus in our protocol, we added 8 microliter. If I go really back here, you see here that this is actually 8 microliter. That's why so according to our protocol, so it's 8 microliter, which is 0.008 ml. 8 microliter of original virus was used, right? So to calculate per ml, so we will convert this microliter into ml. So it will be 0.008 ml, and basically TCID50 per ml will be 2.37 into 10 to the power 6 divided by 0.008, which is 2.9 into 10 to the power 8. This is TCID, okay? This is TCID50 per ml. So this is the TCID50 per ml, but if you want to report this as a PFU per ml, then what you have to do is that you simply have to multiply this number by 0.69, then you will get this number as a PFU per ml. I hope this video was helpful, guys. Please subscribe our channel for more content like this.